Welcome back. We are in general ecology. Today I'll be talking about chapter 11, which is on population distributions. You recall the last set of segments were referring to or looking at life histories in populations, how species, how organisms plan life history, birth, growth, maturation, production of eggs, etc., on into senescence and death, are affected by the environment, okay, and where they live. Today, we're going now into population distributions. And chapter 11 is just about that, and a lot of the material I'll be talking about is in chapter 11. So you'll be able to very nicely go back and forth between the chapter and these notes. And you'll see the figures listed when I use them. By the way, when it comes time to study for an exam or something, your best approach is to go to those figures I talk about in these lectures, and then go back to the book and pull up that figure, read the information, make sure that figure makes sense to you as a standalone image and the material written around that image. And you can kind of help yourself understand it, but then find where it's referred to in the text, and then read that section. Maybe the paragraph or two before, if we're not just covering everything, you can hone in on the figure, read a little bit of the lead-in, really hone in on the figure where it's talking about that figure, and then a little bit on the lead-out, if you will, and that's going to be your best way. You don't have to memorize these figures and the data in them. Remember, these are all case studies to emphasize what we're trying to help you understand about ecology. So, you don't have to memorize the data as much as what did that figure tell me about this particular concept in, let's say, population distributions, right? So the first slide that I'm going to talk about in a minute, we don't have to go forward, but will be on ranges, and that's that first little bullet point there. Anyway, that's a good way to study. So chapter 11 today, population distributions, and that little figure on the lower right shows you a pretty crowded population of some kind of seal, I believe. If it had ears, it'd be a sea lion, but I think those are seal. Looks really crowded, but they like it. That's how they live. They get up there on the shore and just kind of hang out, get warm, go out and fish and do their thing. So that's normal. Now, if humans were in that condition, that might not be so normal, although there are crowded places with humans. And then there are other species that have to be very dispersed. So we're going to look at all of that with different species and how population distributions differ. Geographic range, the abundances of the actual individuals, the density, as that figure shows, pretty dense in that location. Dispersion, sort of how the individuals are spread across the landscape. And then dispersal, different, make sure you get that difference. How they move to new locations, how they migrate to new locations, either within their life history to go off and reproduce, or even with just themselves going out to find new fodder to eat or something like that, right? All right, so first, geographic range. And the example that I like because of the location of where these lectures are being viewed for the most part in New York. And you could see the geographic range of the sugar maple in this image. And probably a lot of you know this, that the sugar maple is kind of all around, but really, where does the maple syrup come? New York, yeah, Vermont, yeah, and other locations, but not down in the southern range, not way down. Once again, I forgot to get my laser pointer. You'd think I would remember this. Bear with me. There, okay, so there's my laser pointer over on the left. Now it's over on the figure. And you don't probably buy a lot of maple syrup from down here. It's not the ideal location. And that's why there's only a couple spots where they've been documented. Even on the lower range of this broad green blob, that's not where you get maple syrup, not down here. you get your maple syrup up here where the climate is perfect for sap and etc., right? But the other thing to think about is that even across this entire range, and even in what we would consider rather ideal locations for the maple syrup production, the trees are not everywhere. You can't just walk into anywhere in Illinois and find a bunch of sugar maple, right? You're going to find certain sort of microhabitats, and that's where they're going to be established. So it's important to realize while you could draw a big circle around where that plant can grow or where that animal exists, they're honing in on very specific habitats within that range. And that's an important thing to remember. For instance, this Clematis fremontii variety Rillier, I think that's how you pronounce it, Rillier. It's a specific variety of a clematis species, Clematis fremontii. Its range is here in Missouri. If you get inside that range, you start to find out that no, it's not everywhere. It's in certain drainages within that range. And then if you blow it up even further, you realize it's not throughout the drainage. It's only where there's really nice soil type that it can survive in. And even if you blow it up further, you find out that there's a sort of clustering of where they occur within that soil type. So I think they're limestone. I think it says right there, limestone outcroppings. So while the range might be the state, very pretty flower, you have to, if you're just going out to find it, it behooves you to find out what are some of the microhabitat conditions that allow that plant to exist in specific areas, and then you'll have an easier time finding it. Just for, straight ahead application, if you want to find that flower, where do you go? Not just anywhere. But the joke I often make is you don't go to the parking lots, right? At least you know to go out into the fields. But then even within sort of the more natural areas, there are soil type restrictions and moisture and etc. Okay, so the geographic range is a very broad brush stroke, but then you have to get into sort of the more microhabitat requirements, and you find out that the distribution is more spotty. All right, another feature about ranges is the density of the individuals. Okay. Or about distributions. It's the density of the individuals, and that varies across a range. And this figure, 11.6, shows sort of a cartoon, maybe a heat map of a particular bird, and you can see that in the whole sort of amber color here, a yellowish color, is where the range of that bird is. But in the middle of that range is where it's really abundant. All right, and that is very common to find a range that's rather extensive, but in the middle of that range, that's where the organism is most abundant. Oftentimes, it's an area where it may have even evolved in this location. That's where it first showed up in the evolutionary history. That's where the genetic diversity is most diverse, right? And then as you get out to these edges, these are birds that get out to the edge of that range, and they're in pretty unique habitats, so not every bird is going to want to get there, can live there, and therefore the genetic diversity starts to drop off out here. I could get into an example of how that impacts or how that impacted human history by talking about potato and potato famine and the blight. When they wanted to find resistance to the blight, they had to go to the range, middle of the range, or the origin of the species largely, to find quite a few resistant individuals or plants or populations, and that was in Mexico, as some of you know, or in the Andes. Not from Ireland, a very limited amount of genetic variation in Ireland because it was dispersed there by humans and only a few genotypes, and they were completely non-resistant to the potato blight. So in order to try to find ones that they could breed back into that population for resistance, they went to Mexico. Okay, so density of the individuals are going to be highest in the middle of the range, and that also affects the genetic. You can also expect greater diversity of genetics in the middle as well. Far more abundant animals or plants in the middle, and the dispersion, they may be more crowded in the middle. As you move out to the edges of the ranges, they're less crowded, they're more spread out individual to individual. All right. Here's an example you may be familiar with. You will by the time, those of you in EFB, by the time you're done, the American chestnut. It's sort of our poster child. Got a lot of work here on resistance. And as you know, the American chestnut came, was here and existed across this range back in the day, right? It can live throughout this whole area, but it got hammered by the chestnut blight, which was imported from China. Bring that back at the end of the lecture series here, how that might have happened. But the chestnut can live throughout, and that's what we call the fundamental niche. The chestnut can grow anywhere from even up in Vermont on the edges, New Hampshire, Massachusetts, all the way down to, say, Alabama, and kind of in an east to west spread as well. So, but they don't today. They used to. The realized niche has been severely limited by the chestnut blight. And if you put a plant out, it will actually, if you put a seed out, it will germinate, or you can put a seedling out, it'll grow, it'll get pretty big if you're in an area that doesn't have chestnut blight nearby. Unfortunately, eventually the spores of the fungus will find it and then it gets killed. And that happens fast enough that the plant will not reproduce, right? It can't get big enough to start making its flowers. And then, of course, you need the other flower type because they need to crossbreed, right? But anyway, the original geographic range, the fundamental niche, where it could survive is quite extensive on the eastern seaboard, well, off the coast a little bit, but limited by the presence of the chestnut blight. And so now its realized niche is actually is extremely limited. You don't see plants reproducing, right? That's going to change now that we have resistance in the system. Okay, so the concept of niche is sort of a new term in the class. That I want you to look up, maybe do a little, not only in your book, but look into your text. Most of you are probably familiar with the term at some level. We think of a species habitat as its home, its niche as its role. I've heard that often. It's not quite like that though. Its niche is also what environmental conditions it needs. So it's a combination. It was a term that was brought into ecology in the early 1900s by a guy who studied thrashers. One of these kind of ground birds, has a nice curved bill. And this was at a time when ecology was just starting to grow. And the first papers come out, beautiful, short little papers. He just sat in a chaparral in California. Of course, there's a California example. And recorded what it did. And from there, we've built this concept. It's a very common concept. I ask you to look on the web for various definitions and things, because it's usually, there's nobody comes up and says, that's the definition of niche. Or if they do, they're sort of not accepting the nuances around the term. So make sure you sort of get a broader understanding of the term. It's not just the role of the organism in the environment or in the habitat. It's the role of the habitat on the organism too, to some degree. Okay. Right. Okay. So now we get into something that I want to spend a little time with, because in the end, we'll be able to come back to some ideas about how an organism might migrate to a new location and get established. And we can look at the sort of separation of species on the landscape. We think that they can get around dispersal, but they're not, right? You don't find them everywhere. There's an old paper from 1930s about microbes on the environment, although microbes were just getting into the scientific mindset, let alone the public. But he said, I'm not translating, I'm paraphrasing a translation, but he said, about microbes, everything is everywhere, but the environment selects. Okay, so the dispersal is everywhere with microbes, all right? Think wind pollination or something. But not everything gets established everywhere because they need certain criteria met, right? So that's what this little experiment allows us to test. The case study is using monkey flowers in California. It's beautiful little, it's kind of a shrubby plant, a lot of different versions, a lot of diversity out there. Not a huge amount, but enough. Anyway, two of the species here, scarlet monkey flower and Lewis's monkey flower. The monkey flower I'm familiar with, when you look at a flower, like this one over here on the scarlet monkey flower, it looks like a monkey face. These don't, that doesn't look like a monkey face to me. Maybe some of you are letting your eyes gloss over and you can see it, but the one that I'm familiar with, and I can't remember its name, but it's yellow with brown spots, grows on the coast, way off on the coast. It has a beautiful monkey face when it first pops open. Anyway, so these are two different monkey flowers, so named for that face. And they're growing in the Sierra Nevada. So here's the San Joaquin Valley, low elevation. If you can see my pointer, I'm on the lower left area of the figure. And then as you move up the elevation, this is the Sierra Nevada, you go all the way to very high elevation. Think of this as Mount Whitney, 14,000 feet elevation. And then it drops off very abruptly into the high desert. Think of this as Mono Lake, and other locations on the east side of the Sierra Nevada. Think of this also as a moisture gradient because clouds, moisture is coming off the Pacific Ocean into the air, blowing onshore, and as it hits the Sierra Nevada, it condenses and cools and forms clouds and drops a lot of rain in this area. And as you move up slope, there's another function, and that is you're getting drier, especially the high desert. By the time you've gotten over here, for this population, it's pretty dry. You're approaching desert type conditions. So it's not just elevation. There's probably soil type variation, although it's all the Sierra Nevada, which is fairly related. Granite block that's been around for a long time and is eroding. But this one is sort of different than those over here because of the moisture gradient that changes also. A rain shadow, look that up if you want to see what I'm talking about there with the water coming across and basically squeegeeing out so that by the time a cloud gets, the air gets here, there's not much water left. So very little rain, the land of little rain. Okay, so back to this. Here's the scarlet. Those are the orange. They do really well. They do well with their annual growth of stems at 415 meters, low elevation on the west slope, and those that survive. Much better than the Lewis's monkey flower, which is better adapted at high elevations. So you can see this is a good reason why. If you walk around in this region, the scarlet monkey flower is quite happy. That's what you're going to see. All right. If you walked around in this region, the greenish area of the Sierra Nevada profile, you won't see, especially out in here, probably here, you're not going to see a lot of scarlet monkey flower. You might, but you're going to see mostly the Lewis's because it does better at higher elevation. And then in this region here at about 1,400 meters, you see both. That's where you'd go to find both. And then there'd be some interesting competition studies because they're pretty closely related. So what are they doing here? Who's eking out an existence in this region when they both can survive and they both put on a fair amount of growth with their stems? Okay. One thing I wish these researchers would have done, and maybe they did, it's just not included in the book. As I said, this is a dry area compared to out here. I wish it may not just be elevation. I wish someone, maybe it's you, would go out there and do another experiment comparing both plants on the east side at 3,000 meters and on the west side at 3,000 meters. And let's see if it's a moisture function. Maybe the correlation here looks like it's elevation, but there may be something more subtle going on with moisture on the two slopes. Okay, so that is, and the reciprocal transplant, what that refers to, and it's a common theme. We may run into more of these later in the semester, and you may run into them. It's when you take a plant that grows in one location and put it in another plant's location, and you take the plant that grows in that location and put it in the first plant's location. So it's a reciprocal transplant. And it is a very good way to look at how these species sort of sort out on the landscape over evolutionary time. Okay. And this leads to something that we call ecological niche modeling, which is pretty cool stuff. I'll explain it and even some applications here in a minute. It's a little bit complicated, but not bad. Okay, so what you do is you go out to a site. Let's say you've got a rare plant, a rare insect, a rare bird, fish. You map the location where that species occurs. So you kind of do your population distribution modeling or distribution assessment, right? We'll explain how you can do that later. But you go ahead and map out locations where they occur. You go back out to those locations and you record the conditions at that spot or in those spots and try to see if there's commonality. If the plant is here and there, is there something going on that tells you, yeah, it needs a certain moist environment, or it only grows in the tropics, for instance, or only grows in the desert, just for, very stark differences there. So that's your ecological envelope. And now you know what that species, or you have an idea of what that species needs. And if you're doing if you're trying to increase the presence of a rare species on the landscape, you might go out and find other individuals and see if they aren't there, you just haven't looked. And so you'll look on other maps and you say, these locations have the same conditions as the ones that have the plant that I've looked at. So I know the ecological envelope. I find new locations where that envelope might exist, and then I do very careful surveys, and I might find new populations of the species on the landscape. And we can extend the understanding of the range of the species, if it's a very rare species, or just in general, this way, by first documenting where it occurs, documenting why it occurs there, and then looking for other locations that have the same conditions. And maybe we'll find more species that way. The other thing that this is really good for is understanding how to avoid invasions, plant biological invasions. So we know what the species needs, and we go out and find other locations where we may not want it, or it doesn't belong, like changing continents, and I'll show you an example in a minute. And we say, wait a minute, we need to be very careful. If this plant grows out here and has this ecological envelope, and then we bring it into the United States, for instance, and let gardeners put it into their gardens, we may be setting us up for a biological invasion, because it will do very well. And it'll actually start moving out into the naturalized, out into the plant communities. This could be insects, this could be fungi, this could be, and all of it, right? Animals. So we look and we see if we can't find areas where that ecological niche or envelope occurs, and then we say, okay, we need to be very careful with that particular species moving into this new area, because it quite likely will become invasive. I've been involved with this in related to pines being dispersed by humans down to the southern hemisphere, where they've never lived until now. And they're quite invasive. Okay, so here's an example from your book. It's the ecological niche modeling on Chinese bush clover, which occurs in this region of Asia and up into Japan. And you sort of see the projected range. It will do very well where it's dark, and it can actually get established where it's light colored blue. Now I'm on the left panel, panel A, So this is the native range of this plant. And when they did the ecological niche modeling on this area where they were occurring, they say, well, looks like we might have a problem in North America because the same conditions occur out here. And in fact, now that plant did get over here. This is before people were really concerned about invasions, biological invasions. That's sort of a new idea. And now they've looked at it, and now this Chinese bush clover is all over the place. And you could tell, again, the known distribution over here, all the purple is pretty heavily colonized. Even out in California and up into Washington and Idaho, Montana, it's out there as well, and down in Mexico. And it could be predicted by the ecological niche modeling. But it's become quite invasive here. It didn't become invasive in California because it needs a certain moisture level, which it gets in the farming belt of the San Joaquin Valley. But when it gets up to the Sierra Nevada, it loses that part of the niche that it needs, the moisture. Here, it's all agriculture. It's all what's the word I'm looking for? They're using water to grow the plants. You guys know what the word is. I'm just drawing a blank. Anyway, that's ecological niche modeling, and one can use this to avoid invasions. With this information, they might have said, okay, gardeners, you're not allowed to plant the Chinese bush clover because we don't want it to invade. Okay. Here's an example of the ecological envelope, that is sort of the niche, the envelope where this group of fish might exist, in this case based on temperature. And we're in the North Sea now, all right? So this isn't really an invasion of fish into this area. These fish were there. And what they found is that the North Sea, because of global warming, temperature changes, at the bottom of the North Sea, the temperatures have been going up since 1975, all right? And you can see the data points and the spread of the data and the regression line here up to about, I think the last data point was probably somewhere around 2003 or something. So the temperatures are going up, but it turns out the fish communities are responding. And what they found is that as the years progress, all right, so here we had temperature, this panel up here on the upper left, we have years and then temperature on the y-axis. At the same time, down at B, we have years, just like the x-axis, but now on the y-axis, number of fish species collected. And that's going up. So diversity is going up in the North Sea of fish as this time goes on, and it's directly correlated, it would appear, by the temperature going up. And the theory is, as the temperature goes up, if you're collecting up in the very north of the North Sea, there's only certain species that would be living up there because it's rather cold. But as the temperatures have gone up, the fish that were down here, where it's warmer, are migrating north with that higher temperature, and therefore overall the diversity is going up because you get a mix of these, both cool and warm water fish. That's my understanding. So again, something we could predict using the ecological envelope of the different species. And of course, this is something that's sort of a story in progress. You might go out there and you say, boy, there's great diversity in the North Sea. This is great. We're doing the right thing. They're sorting it out. They're starting to duke out who gets to win where. And if you were a researcher and you weren't taking into account the temperature changes, you might get a very wrong impression of why that diversity is there and start doing some sort of, policy changes. Maybe all you need to do, and increasing diversity, but in fact, you've got a snapshot here, and it's going to be shifting a lot. The Adirondacks are not different, by the way. We have things like beach bark disease up there that's changing things, and if you just go out there now and measure your trees, the species and the communities, it's directly impacted by beach bark disease. beech bark is there, beech is there, and other species are starting to compete in different ways because of those two features. There's another thing that happened not long ago, a lot of logging, where humans got up there and started harvesting certain tree species, and then only the big ones. And so the sort of less fit, less adapted individuals were left to reproduce. And so we see all these infestations in part because humans are bringing the insects and the fungi and such in, but in part because we left the non-resistant individuals behind. And we took the big trees for our purposes. And so if you go out there today, you say, this is a beautiful forest, nice and diverse. It's still shifting. There's a lot of shifting going on as to who's going to dominate where, and the communities are going to reflect that. So always good to keep in mind the whole picture in a system as you look at species distributions. Right, okay, and so I'm going to shift gears now to how we look at the dispersion of the species on the landscape and ways to do it, but also what patterns we see and why they're there. So first, the patterns we see. These are, of course, sessile. Well, the penguin is not sessile, but doesn't really move as much as, say, an albatross, right? Even though it's a bird. Its wings, not really good for flying, better for swimming. But then it comes back on to the shore and makes its nests, and they are very evenly spaced, right? Okay. Up above, we have a mushroom species, a Hypholoma fasciculare. Don't worry about that. Those here, it's a clustered distribution, and we could look at why you would get a clustered distribution on the fungi. And then in the lower panel is a random distribution, and that's dandelions. You've seen that. And there's really, some are quite clustered together, some are more dispersed and separate from each other, and what we call random. So the three panels are clustered, evenly spaced, and random. Now, looking at this a little bit more, if we go up to the top panel there, yeah, good, that clustered. Again, those are fungi. It's a little bit weird to use as an example. The editor tries to bring in mushrooms periodically, maybe because I've been queried a few times to help with making the text better over the years, and I'm always saying, there's some examples that might work. He should have consulted me here because those Hypholoma are probably one individual, and there's a mycelial network in the wood, and parts of it are fruiting and parts of it are not. And when they fruit, it's just a bunch of flowers, right? Those are not individuals fruiting. That's one individual fruiting in clusters, and that's not exactly what we're talking about with dispersion. It's the dispersion of individuals on the landscape. But I'll show you a better example in a minute. Okay, so let's go ahead and move on to a better example. Here's a cluster distribution of aspen. I believe this is in the Rocky Mountains. And individuals are distinguished by the timing of their leaf fall, or even the color. So there we go. Looking here, this is crazy. You ready? This is one clone. This is one individual of the aspen. All right? And in the fall, its leaves turn color, and they're going to drop off. So underground is a root system that puts up stems. All right? We're used to seeing stems above ground coming off the tree bole. But here in this species, the roots are underground, and the stems just come up and look like individual saplings or trees. You can see that here, this individual has already dropped all its leaves. And each, what we would think of as a tree, is just a stem off the root system below ground. And then here, so there's 3 individuals here. Well, maybe 4. All right? And yeah. So it's clustered. You've got these 4 individuals kind of on this ridge, and then you could see some other individuals clustered in areas around in this whole landscape view. right? And some of them are quite close together. Some of them are dispersed with some more distance. And so that's a clustered distribution. So the question is, why? Now, in the mushrooms, I told you that's just one individual mycelium, and it just happens to fruit in locations. It's not really a cluster. It's one individual. It's not fair. But in this case, with these aspen, and the beech is the same way, by the way. You could go out to a beech forest, you post beech bark disease, and you get all these little stems coming up. The big mama tree has been killed off, but then you get all these stems coming up. Those are all the same genetic individual. So sometimes, well, that's one reason why you get clustering, that it's actually just one individual. And until we had genetic techniques or very careful researchers, we didn't know that those stems are all just that same clone individual acting as an individual, dropping all their leaves at one time. I'll come back to that. Here's another dispersion pattern, and these are evenly spaced. The penguins was the example that the book uses, but I like this one from the desert. You have these little shrubs. They're not huge because it's really dry. But out in here, there's a nice evenly spaced distribution pattern. Now note that maybe where the picture is being taken from here, it's getting a little bit more crowded, right? They're still kind of evenly dispersed, but they're more crowded than out in here. And then over here, it happens again. So ask yourself why. What do you think is driving this pattern? The other hint I've already mentioned is that this is the desert. All right, so if you've thought it through, or maybe you've been out in the desert, or maybe know that a desert doesn't have a lot of water, these plants here, they're all basically getting established and either dying because they can't compete against the others for water, or they can. And when there's a little bit of a drainage depression here, you get more water, and maybe that's what's going on here. Therefore, you can get more plants. So the same process, but the issue, that is water, that they're competing for, changes across the landscape. All right, it kind of looks like a clustered distribution pattern or dispersion pattern, but I think I want you to be careful with that. These are all under one condition, and think of just draw it up here. I should maybe make this figure tighter so that that's all you're looking at, and you can see that they're relatively evenly spaced, and I'll come back to and to why in a minute. Okay, and then lastly, random. And I pulled this up from some slides that I had. This is a little fungus, of course. It's growing on a log that's highly decomposed, and there's a lot of moss, and the distribution pattern is random. Now, unlike the fungus that the author of our book used, this one, each one of those is probably its own individual. They don't form big mycelial networks. And you can see that basically some of them are very close together, and some of them are widely dispersed. And on, on a micro scale, widely dispersed. So this is a function of spores landing. There's no other factor. If they land and germinate and start a new individual, they'll fruit, and then they'll die. And that's a random distribution pattern. There's no real influence driving that pattern other than it's, should I dive into my Southern California? Dude, it's random, okay? Anyway, you get the point, random. So in each of those slides, I've asked why, and I've kind of hinted at some of that. Here is a cartoon from, 11.7, showing the clustered, random, and evenly spaced. And I want to give you sort of the mechanisms or causes behind these three. All right, so I've already asked you, and I've kind of given you some hints on some of these, but now we'll have it written. So the clustered one, this happens when you have social interactions. So maybe organisms, maybe these are packs of wolves in different drainages, and they are clustered. They occur together. There's very little dispersal to the other drainage, probably because the other drainage has a group there that will discourage it. But it can also be plants like those aspen or the beech, where an individual gets established and a couple others, and they're all producing their stems around those individuals, and you get that clustered. A new one might get established out here, hasn't had time for the root system to form and all the little stems to pop up out of the ground, so it's all on its own. These might be much older. This one might be, 30 years old. This is 500 years old. So there's more stems in that situation. Okay. Also resource patches. So maybe there's more water here. And if we backed off from that one desert image, one could say, okay, that's a clustered pattern because there's water in some of those depressions in the ground, and therefore you get more plants there. But I wanted you to focus on the middle zone where they were all evenly spaced with one resource patch or one resource amount that is water in a homogeneous level, not heterogeneous. Okay, and then it can also be local dispersal. So plants sometimes have, I'm thinking plants here, seed that blow off the seed capsules, and around that individual, that mother plant, is where you get all the new plants getting established. Or you can think of something like a quercus dropping its acorns. They don't go far, right? Unless people or other animals move them, like blue jays and things like that. But for the most part, they all land right there, and that's where you find most of the seedlings. So that is local dispersal. The vegetative spread I put in parentheses, that's my beech or populus or aspen example, vegetative spread underground. All right, so random, or did I go next to evenly spaced? Let's see, random. So here is a place where there's abundant resources. That log with those little antler-looking fungi, tons of resources. If they land and germinate, they're in a good spot. The microhabitat is good, it's moist, it's got moss there to help keep things moist. It's a good spot. And just randomly, you might get a bunch that get established in one location, but only one out in here, and so you get a random distribution. The resources are homogeneous across this area, all right? That's a good thing. And then you get ubiquitous dispersal. So the dandelions, they could just, once they put out those little wind-blown seed, they go everywhere, right? And then they get established, and you looking into a lawn that has not used a chemical to get rid of them, you'll see that random distribution when the flowers pop up. And finally then, evenly spaced. This is all about competition. So in that central region of the desert image I showed, those were evenly spaced. The roots, the seed got there, it germinated, made root systems, and if it was lucky, it got established, produced roots, and grew out until they ran into the zone of another one. And when they ran into the zone of another one, the water resource then was cut off because the other one is sopping it all up. So they become very evenly dispersed, probably a function of the amount of water around and how much that species needs, and how big the root systems can actually grow. And what we see above ground is not what we see below ground if we look carefully, but that's for people that look at roots to discuss. So evenly spaced is competition. And you can think of other examples of animals that are evenly spaced on the landscape. So that's it for this segment. In the next segment, I'll get into how we actually are measuring these things. You just don't go take a picture and count them or make a cartoon like we've been looking at. We have very specific ways, quantitative ways, to assess the dispersion on the landscape. So we'll go ahead and do that in the next section. Thanks, and may the rest of your day be good. Cheers.